Good evening to everybody. First of all, I'm presenting myself. I am Roberto Chiarle, an associate professor at Harvard Medical School and a research assistant at the Boston Children's Hospital. And I'm going to talk tonight to you about some recent advances that we made in our lab about novel therapies that can be used potentially in the future to target ALK mutated, in particular ALK-rearranged cancers. And I would like also to thank Bioconference Live to grant me this opportunity to present my work in front of you as an audience. Even if I don't see you as an audience, I will be very happy to present. So the work that I'm presenting tonight starts with the idea of boosting cancer immunotherapy for a specific type of tumors. And as you might know, the cancer immunotherapy relies on the recognition by the immune system of cancer cells. And this recognition is a very long process that requires several steps. And in these slides, there are summarized some of the key steps. According to this slide, there are like 7 steps, but they can be a little bit more. But in any case, the very first step, he starts with the release here in point 1, with the release of antigen specific from cancer cells. And typically cancer cells, they can have specific antigens, either because they carry chromosomal translocations or because they have somatic point mutation in protein that turn out to be antigenic and be recognized by the immune system as antigens. So this is the first phase in which a dying cancer cell is kind of releasing antigens that are novel for the immune system, and they can be captured, and this is phase 2, they can be captured by the cell presenting specialized cells that present the antigen, typically dendritic cells. And these dendritic cells or several types of macrophage then can eventually migrate to lymph nodes. And here is point number 3, in which after migration to lymph nodes, at that point there is the real happening of the immune reaction in which presenting cells, they carry on the antigen and present to T cells in the lymph nodes, and also B cells, of course. So there is a strong interaction between presenting cells, B and T cells, and also other cell types like Treg cells, Th17 cells, in the lymph node where the immune reaction really takes place. After the antigen is presented by the specific presenting cells, and the T cells or B cells are primed to recognize the antigen, then the next phase is trafficking. We are talking mainly on T cells because cancer recognition is made mostly by T cells. But, I mean, of course, also B cells are important, but most of the anti-cancer activity of the immune system goes through T cells. So in the stage 4 of this immune reaction, now these T cells that have been primed to recognize specifically cancer antigen released by the cancer cells, now they go back to the blood vessels, they go back in trafficking, and they basically move around the entire body, and then eventually they can infiltrate inside the tumors. And here is phase 5, in which T cells, they leave the bloodstream, and then they infiltrate the tumor, and there in the tumor site, so this is phase 6 and 7, basically there is a first step of recognition of the T cells, the prime T cells in the lymph node now recognize the antigen, the same antigen now presented of the cancer cells. And finally, if everything goes fine, finally there is the last step, that is the killing process, which is the ultimate goal of the immune system against cancer. So you see, it's a very long process that requires a lot of components, and you see highlighted in green and in red a lot of factors that can affect this process either by stimulate and increase the efficacy of the presentation and priming and migration of T cells and T cell recognition of the cancer cells. But there are also, I would say, an equal amount of factors that inhibit this process. And all these factors typically are inhibitory factors that work in the immune system to block or prevent an exaggerated immune response against bacteria or viral antigens. So basically you see there is a long list of pro-cancer factor and anti-cancer factor that can affect the balance of this entire system. Why and how we can manipulate the immune system to target cancer cells? So of course, cancer vaccine or cancer vaccination is a topic very important in the past, I would say 3 to 4 decades, maybe even more, in the battle against cancer. And there have been several experimental ways to use and exploit the immune system to recognize better than it does naturally the cancer cells and eventually help in the killing and elimination of the tumor cells. So you see here in this recent review, summarize the most important, I would say most effective way to target cancer cells that have been developed throughout the year. So there have been vaccine trying to use irradiated cytokine releasing tumor cells, in which the cancer cell, they release both the cytokines and antigens, or also dendritic cells that can be primed to present antigen to the immune system. And these cells, they can be infused into the patient to activate the dendritic cells with specific antigens that are, of course, tumor antigen. And this priming results in an activation of tumor-specific CD4 and CD8 T cells that are the same cells that eventually recirculate and find the tumor and kill it. Another way is to exploit in situ the release of antigens by cancer cell through either apoptosis or through other way to kill cancer cell, like drugs or chemotherapies that can kill and help the release of specific antigen from the cancer cell. So this first set of tools basically deals with the idea of providing more antigen to the presenting cells so the immune system can have more antigen and work more efficiently. The second way to help the immune system to recognize the cancer cell is to modulate the interaction between the tumor cells, which is here, and the effector T cells, which are here. So you see that typically this interaction is mediated by a specific recognition of the T cell receptor of the antigen presented by the cancer cells. And this effector function can be modulated either by agonistic co-stimulatory molecule or by antibodies that block immunosuppressive molecule, typically CTLA-4 and PD-1 ligand the system. And we will talk about this, the potential therapeutic use of these tools later on in the presentation. And finally, there is a third way to modulate the response of the specific tumor cells, which are here, the specific T cells against the tumor cells, which are here. And it is the way to manipulate or eliminate cells of other lineage, either myeloid suppressor cells, like here represented in the corner, or Treg cells, which Treg cells typically inhibit or dampen the activity of specific anti-tumor cells or T cells in general. And so there is a possibility to manipulate or to eliminate or to change the function of this either regulatory or suppressive cells to increase the efficiency of T cells to recognize the specific cancer cell and then lead to tumor destruction. So you see there are many ways to manipulate the immune system for an increased recognition of the antigen presented by cancer cells and to increase the efficiency of specific T cells to recognize and destroy cancer cells. So we decided to work on this aspect on the immune system because we have been working for several years with a specific oncogene which is called Anaplastic Lymphoma Kinase, or ALK, which was cloned about 20 years ago in a specific category of lymphoma called anaplastic large cell lymphoma, and in 1994. And this ALK is a tyrosine kinase that is translocated, as you see, with several different partners in lymphoma. And as a result of the translocation, for example, of the most common NPM-ALK translocation, you see that many of the tumor cells, in this case lymphoma cells, they start to express the translocated oncogene. So here is an immune reaction against the ALK detecting the fusion protein NPM-ALK, and you see that the antibody strongly reacts against the cancer cells, like this one indicated by arrows, but many of the normal cells that surround the tumor, like this one, are totally negative for the protein. And this is easy to understand because ALK is normally not expressed in normal T cells or B cells or lymphoid cells, and we say it's very low expressed in any normal cells of the human body after development. Only few neurons retain a significant expression of ALK as a wild-type receptor on the surface. Instead, after chromosomal translocation, the expression of ALK, which is only the cytoplasmic domain, is driven by a different promoter, NPM in this case, and the result is a strong overexpression of the fusion protein that can be detected by antibody against ALK. And you see that after the discovery of anaplastic large cell lymphoma as the first tumor to have ALK translocation, then several other tumors were demonstrated to carry similar translocation. For example, inflammatory myofibroblastic tumor, in which about 50% they have, you see a wide range of different ALK translocation, and most recently and importantly, non-small cell lung carcinoma, breast cancer, and colorectal cancer, as well as renal carcinoma. So another typical example is non-small cell lung cancer, in which the most common translocation is the ML4R translocation, and you see here depicted non-small cell lung cancer with an immune reactivity against with an ALK antibody, and you see again that the cancer cells that in this case they cover the alveoli. You see here there are all these surface of the alveoli that are covered by cancer cells. You see that there's again positive for ALK. I would say less strongly than lymphoma, but still positive, whereas all the other cells, like here the stromal cells, endothelial and lymphoid cells, they are negative for the ALK expression. So we thought that this window of expression between cancer positive cells and all the other cells that are negative could be exploited for cancer vaccines, so to instruct the immune system to recognize specifically this ALK protein that is expressed by these cancers and prime the immune system for a more efficient anti-cancer response. So what are these very peculiar biological properties of ALK as an onco-antigen? And you might notice that we use the word onco-antigen here because it's not a simple tumor-associated antigen, but is an antigen that is expressed by cancer cell, but is also oncogenic. And so what are these key immunological properties? First of all, as I already mentioned, in normal tissue, the expression of the wild-type ALK is very low and is typically restricted to some neurons in the brain or in some motor neurons. And so there is very, very limited, if any, expression in the vast majority of normal tissues. But then, after chromosomal translocation, there is a selective and high expression by the tumor cells. So as I said, this window of expression, actually this gap of expression between between tumor cells and normal tissue is very important to facilitate the immune recognition of the cancer cell by the specific T cells. And these 2 points that follow are also very important. So there is a strong addiction of the neoplastic cells on the ALK signaling for the growth and survival. And that's very important. That goes back to the meaning of onco-antigen. So it's not just a simple antigen expressed by the tumor cells, but is an antigen that is required by the tumor cells for growth and survival. So the important point of this statement is that typically cancer cell cannot survive without the expression of this antigen. And that's very, very critical because most of the failure in the past of typical cancer vaccination strategy were probably due to the selection of clone inside the tumor that no longer express the antigen. So even if the immune system was primed well against antigens, then some clone escaped without the expression of the antigen anymore, and there was no way for the immune system to tackle them. So in this case with ALK, I would not say that it's not possible, but I would say that it's very, very unlikely. And to confirm this point, there was a recent study by a group here in Boston showing that 100%, so it means all the tumor that relapse after specific kinase therapy, I will talk about this one later, all the tumor, 100% of the cases, they still express ALK as an antigen. So this is very important to plan a strong immunotherapy against this tumor. And finally, also very important is that the ALK protein itself is a very, very good immunogen for the immune system. And that's important because some antigen expressed by tumor could be perfect or could have several important features, but then eventually they turn out to be a very poor immunogenics for the immune system. So even if you try to vaccinate, the immune system does not recognize those antigens. In the case of ALK, there are evidence that ALK is already a spontaneous, very good immunogen. In fact, all the studies were made in patients with lymphoma, and was demonstrated that there is a spontaneous anti-ALK immune response in patients that carry ALK-rearranged lymphoma, like the MPM ALK that I was showing before. So typically, these patients show circulating antibodies against the fusion protein, mostly against the ALK portion of the fusion protein, very little, if any, against MPM, because MPM is a constitutive express protein that is present in every cell. So they typically develop antibodies against the ALK portion of the fusions. And this antibody, the highest is the titer of this antibody, the better is the prognosis. So there is a link between the intensity of the immune response and the prognostic of the evolution of the disease. But these patients, they also develop specific T cells, memory T cells with cytotoxic potential, so like CD8 plus T cells, but they also develop CD4 plus T cells. And what is most important, it was recently demonstrated that the combination of this immune response by T cells and antibody can stratify this patient into two different prognostic groups, which means that more or less all the patients, they have this immune response, but the patient that they have a stronger spontaneous immune response, they have a much better prognostic survival and prognosis overall. So I think that these are evidence in vivo, in human experimental, I would say, models that ALK itself is recognized by the immune system as a foreign antigen, is presented, and it generates antibody or a specific T cell response against the protein. And this immune response already has some effect in containing the tumor growth. So we were really primed by this data to plan a way to boost even further by a specific vaccine the immune system to recognize ALK as an antigen in different type of tumor, and I'm going to present data against lymphoma and against lung carcinoma. So we decided to use as a first strategy an ALK DNA vaccination, in which we generated a DNA vaccine that encodes for the cytoplasmic portion of ALK, exactly the same portion that is translocated in all those fusions that I listed above. So by this DNA vaccination, we can inject the DNA subcutis or intramuscularly in the mouse, and there will be an inflammatory reaction, and then this DNA will encode for the ALK protein. The ALK protein will be presented as a single peptide to the immune system, and a specific cell-mediated cytotoxic response will be developed by the organism. In this case, all the studies are done in the mouse models. So DNA vaccination is very effective. And indeed, we show that a simple, very simple protocol of vaccination in which there is one priming vaccination with DNA at the day 0, so at the beginning of the experiment, and then a second vaccination that works as a boosting. They are efficient enough because when you challenge the mice after another week, so you allow the time to build up a strong immune response, then you can challenge the mice with ALK-positive cancer cells, and the results were pretty positive in the sense that we could show that by DNA vaccination, first of all, we were developing a very strong ALK-specific immune response. And the way to test this specific immune response was to evaluate the cytotoxic activity in vivo, in mice, by an assay that is based on a CFSC labeling of 2 T-cell populations. So here you see on the left, these are CD4 T-cells, okay, that are wild type, so they do not express the ALK antigen, and instead here are normal CD4 T cells that express the ALK antigen. So we derive these T cells from our T cell transgenic mice that express the NPM-ALK fusion specifically in T cells. So in both these populations are normal T cells. The left population is wild type, and the right population is identical except that express ALK. And you see that in a control vaccinated mice, the two populations are pretty equal. But in an ALK vaccinated mice, there is a selective depletion of only the ALK T cells, whereas the wild type T cells here on the left of the ALK vaccinated mice, they are practically unchanged. So this is a very nice assay to demonstrate in vivo a specific cytotoxic activity against normal T cells that express ALK, whereas the wild-type T cells, they are untouched. And you can measure that, and we measure that the cytotoxic activity was depleting between 70 to 80% of the cells that we injected, so it was a very strong activity. And you can also track in the spleen by injecting ALK-positive and ALK-negative T cells, and then do a specific staining for ALK, and here you see that there are, in control mice, there are few cells that are ALK positive because this is a tail in vein injection, so there are few T cells that home to the spleen, and you can easily recognize by a specific staining. Again, you see the majority of the normal splenocytes, of course, are ALK negative, but in ALK vaccinated mice here on the right, you see that the number of specific T cells only in the spleen goes almost to 0, definitely below 10%. So you can also evaluate this way the specific cytotoxic activity against ALK positive cells. And we show that by this protocol of vaccination, the protection of the mice, so we first vaccinated mice and then we injected the lymphoma, which was ALK positive, was really strong. Actually, all the mice, 100% of the mice, were protected when we vaccinated with a specific ALK vaccine. And this is a number of protections, so you see that there is no protection in untreated mice or in mock vaccinated mice, so it means that all the mice die because of the lymphoma, no protection. Instead, 100% of protection in ALK vaccinated mice, and we also vaccinated with a mutated construct that doesn't have the kinase activity anymore, and still the protection was very high. And what is important that this protection was very long-lasting because in some experiment we vaccinated the mice exactly in the same protocol, so 2 DNA vaccine within 2 weeks. And then we just waited for 7 months, and then we challenged the mice after 7 months. And you see that the protection is still very high. It's not 100% anymore, but it's still 70%. So it means that even with one single round of vaccination, you can obtain a very long-lasting immune protection. And here you can measure directly inside the tumor by injection. So you see, for example, that we, in this experiment, were injecting lymphoma cells, ALK-positive lymphoma cells in the flank. And here you see with the arrow, there are the inguinal lymph node here, okay? Instead with the asterisk, there is the tumor. And you see that in control mice, mock vaccinated mice, you see that the tumor after 7 days is as big as the lymph node, but then eventually grows much more. And after 3 weeks is a large mass of the lymphoma cells, are positive. Instead, in the ALK vaccinated mice, you see again the lymph node with the tumor at day 7, they are more or less the same size, but then eventually the tumor is completely gone and you are left only with the lymph node. And you can see here in immunohistochemistry that in mock vaccinated controls, there are a lot of ALK positive cells. Instead, in the ALK vaccinated, there are the majority of cells are negative, reactive inflammatory cells. And here below, you cannot see very easily the immunofluorescence staining here, but you can, we could demonstrate here, to be faithful, that in ALK vaccinated mice, there was a significant increase in cytotoxic T cells as tested by granzyme B and perforin positive cells. So there is a specific immune response against lymphoma cells that prevents tumor growth. And this is true also when we injected not in the flank but systemically ALK lymphoma cells. So here in the right there is an experiment in which we vaccinated mice either with control or ALK vaccine, and then we injected with ALK positive lymphoma cells. And you see that all the control died very fast, but most of the ALK vaccinated mice were protected, and actually they survived for a long time. And we could also show that we could efficiently combine some current therapy. For example, lymphoma are treated with chemotherapy, so we could safely combine chemotherapy and ALK vaccine, which is this curve here, to obtain an even further increase of survival of the mice. So potentially we show that we can combine vaccination with chemotherapy. And finally, a little bit of mechanism. Here again is in this case is a tumor growth. So we injected the ALK positive lymphoma cells in the flank, and you see that typically control cells, they grow very well and fast. Here are control cells. And if you vaccinate mice and you deplete for either CD4 or either CD8, you see that with CD8 depletion, the tumor grows as the control, whereas with CD4 depleted, which is here, the tumor, they grow much less. So it means that CD8 are really required for the cancer activity, the anti-cancer activity by the immune system. Instead, CD4 are less important. And by doing the same experiments in interferon gamma knockout mice, in perforin knockout mice, and in B-cell knockout mice, we showed what is more important is that interferon gamma is totally required for this, as expected, for this immune system to react against the ALK antigen against lymphoma cells. Perforin is partially required. Instead, the production of antibody, which means the presence of B cells, is totally dispensable because, as you see here, the tumor was as protected, as well protected with or without B cells. So B cells are really not needed in this strategy of tumor vaccination. So what we learned from these lymphoma studies, we learned that the ALK vaccination elicits a very specific cytotoxic immune response that specifically target ALK positive cells. And mechanistically, CD8 T cells and interferon gamma production is essential for the vaccine to work efficiently, whereas B cells or antibody production are dispensable. And the ALK vaccination is long-lasting. I mean, up to 7 months, there was still a very good efficiency. And the efficiency or the efficacy of the ALK vaccination depends on tumor burden. I didn't show data about this, but the smallest is the tumor, the most efficient is the ALK vaccination. And the vaccination is very strong in prevention, so there is 100% of prevention of lymphoma grafts, but is also synergistic with chemotherapy, is potent alone as synergistic with chemotherapy in a therapeutic protocol of anti-lymphoma vaccination. Okay, so in the second part of the talk, we switch from the lymphoma model to another type of ALK rearranged cancer, which is non-small cell lung cancer. And non-small cell lung cancer rearranged for ALK have become a very hot topic recently in the cancer field, because there was the first chromosomal translocation recognized in lung cancer, and one still of the few. There are a few other clones, but this was the first one. And you see that this translocation, again, like in lymphoma, fuses typically the ML4 gene, different exon. You see that the length of the ML4 fusion can be different depending on the different type of translocation, but the ALK portion in this fusion is always almost identical. There are very, very few amino acid changes. And even if you study different type of translocation, like the TFG-ALK or other type of translocation which are rarer, still, again, the translocation happen to be exactly the same in all lung cancer patients, and which is exactly the same as in the lymphoma patient that we just showed before. So we decided to try to vaccinate these mice, but of course we first needed a model. So we built up a model of lung cancer expressing the most typical translocation, which is ML4 ALK, and also a second model expressing the rare TGF-ALK. And the specific expression in the lung was achieved by introducing the fusion protein under the promoter of the SPC. SPC is the surfactant protein C, which is expressed by the alveolar type 2 lung cells. And you see here that by quantitative PCR reaction that only lung really express significant amount of the ML4 ALK fusions, where most of the other organs were negative. And importantly, the levels of expression by Western blot, either in the ML4 ALK transgenic here or in the TFG-ALK transgenic here, they were quite similar to the expression level in human cancer. Here on the left, there are two human cancer cell lines derived from human lung cancer, and you see the expression of these two fusions. The ML4 ALK is the highest and the TFG is shorter, but is comparable with the transgenic expression. And also by messenger RNA expression, we demonstrated that the level in our transgene of the fusion is comparable to the expression in cell lines, and of course also in primary tissues. So we thought that this model was pretty good, and indeed these mice, they develop quite fast. After 4 months, they started to develop tumors in the lung. Here you see this mass that are much more clear and white compared to the pinky lung parenchyma. And you see that by histology, there were multiple tumors here that would turn out to be quite strongly positive by ALK. But again, you see that the expression level of ALK in this tumor is comparable to the human tumor. You see here is a human non-small cell lung cancer. You see that sometimes in human patient the expression could be lower, but in many cases this is the range of expression, and you see that in the mouse we had a similar level of expression. And the beauty of this system was that we could follow up the tumor development directly in the lung, in living animals, by doing MRIs, which is nuclear magnetic resonance. So you see here that after 2 months, there are few tumors appearing in the lungs, here these white spots, okay? And after 4 months, like in the histology picture, now the tumor are much more. They are different masses, they are confluent, and eventually the mice die. You see the survival curve of both transgenes. You see that 100% of the mice die between 60 weeks of age, with a mean survival of about 35 weeks. So we thought that this was a good model to test ALK vaccine against lung cancer, so we decided to go with DNA vaccination. Again, in this case, since these are primary tumors and not grafts, we decided to do 3 rounds of vaccination. And the important thing is that before the first round of vaccination, we did an MRI to score the number and the tumors before treating the animals, so we could really group correctly the mice. And after the first boost, we were doing cytotoxic in vivo assay, like in the lymphoma cells, and then we were following up mice with at least 3 rounds of MRI over time to check for the number of tumor and the size of the tumor growth. And you see here that the cytotoxic activity was very strong. So here are mice vaccinated with a control plasmid. You see that there is less than 20% cytotoxic activity, probably due to a spontaneously cytotoxic activity against the ALK protein. But after vaccination, we really could boost up the cytotoxic activity again back to more than 80%, like in the lymphoma model. So we were very, very pleased to see this strong boosting of the specific ALK response in lung cancers. And we check and measure by MRI the development of tumor, and we could show that in vaccinated mice, here in black, the number and also the size of tumor was clearly reduced by the vaccination as compared to control, which is this curve here. So you see there is a huge difference in number on neoplastic foci. And also the survival was clearly extended between the two either control vaccinated, here in white, or the ALK vaccinated mice, and you see that there was a significant shift of survival. We did not cure the mice, but we had to take into consideration that, as I showed here before, in this transgenic system, there are really several tens, even hundreds of tumors that develop over time. So it is conceivable that the immune system can tackle them at the beginning, but eventually over time it is overwhelmed by the new tumor that keep growing. If you see in this MRI, you see how many tumors they really fill up the lungs in 4 months. So in this condition, the immune system has really difficult to act. So, but still, even in this condition, the gain of survival was very significant. And when we observed the tumor by histology and immunohistochemistry, we could clearly detect an increase of T cells, effector T cells inside the tumor. So here you see control vaccinated mice in which the tumors in the lung are quite big and large. And if you stain for CD3, so for the presence of T cells inside the tumor, you see that there are very little cells spontaneously present inside the tumor. But after vaccination, you see, first of all, the tumor size is much smaller. And second, you see also the color changes because the tumor is filled up of very small dark cells, which are, of course, T lymphocytes because they are strongly positive for CD3. And you can count them, and you see that there is a huge increase of CD3 T cells inside the tumor. So this really means that we vaccinated the mice in the muscle, in the leg, and these specific T cells, they travel in the bloodstreams, they go up to the lung, and they recognize the presence of ALK antigen in the lung tumors, and they start to the cytotoxic activity and kill the tumors. So we really thought that it was the demonstration that T cells were attracted by the cancer cells, and they were very active. But as a downside effect of the ALK vaccine, we also observed that together with effector T cells, we were also increasing Treg cells. Treg cells can be clearly stained by FOXP3, which is a nuclear staining. You see here all the Treg cells that populate the tumor, whereas in control only few Treg cells. So by tumor vaccine, we could increase the number of effector T cells, but also Treg cells in the tumor. So we decided to go back to this strategy, so to deplete Treg cells. Remember this cartoon that I showed at the very beginning, so deplete T cells and see if we could boost the vaccination. And indeed we could, because when we were comparing vaccination alone, which is here, this is a longer time as if, the one that you see before, with a protocol that combined vaccination and depletion of effector T cells, you see that the combination of vaccination and effector T cells were more efficient in preventing tumor growth, and also there was a further gain in survival. So this is a potential way to boost the cancer vaccine against ALK. But you might know that one of the major breakthrough of the past 2 years against ALK-positive tumor, and that's why it is a very hot topic, is that this type of tumor can be very, very efficiently treated, at least for a certain window of time, by a specific ALK kinase inhibitor, which is called crizotinib. And these are recent publications that show that really crizotinib is effective. Here is a human case in which you can see by MRI or by TC scan this tumor infiltrated the lung, and after crizotinib, this tumor almost completely disappears. So this treatment is very powerful. The problem with this treatment is that over time, crizotinib is no longer working because the resistance is happening. I will talk later why this resistance is happening. So eventually the tumor growth comes back, and you have again in a tumor growth that is growing back. So sorry, there is some noise from the phone. And so there is a gap of window in which this ALK inhibitor is efficient, which lasts for about 6 to 9 months. But eventually, after this gap of window, the tumor comes back, and eventually the patient will die. So we decided to use this gap of window in our mouse model. So first of all, we checked if the mice were sensitive to crizotinib, and if crizotinib could be combined safely with vaccine. So in the first experiment, we were combining, as you see here, crizotinib in the middle of the first and second round of DNA vaccination to see if the crizotinib treatment was affecting the response of the immune system. And the answer was no, because you see here the control, and when you combine vaccination with crizotinib, you see that you get as much as good cytotoxic activity as the vaccine alone. So crizotinib could be safely combined with ALK vaccination. And next, we wanted to test this combination in vivo in mice. So if you look here at the left panel here, you see in crizotinib alone treated mice, you see that at 4 weeks of age, some tumor they start to develop, here indicated by an arrow. You treat them with crizotinib and they completely disappear. But when you stop the treatment with crizotinib, and we did that to mimic the relapse of patients under crizotinib treatment. So when we stop giving crizotinib to the mice, eventually the tumor relapsed exactly in the same position, and new tumor came up. But importantly, when we did the same protocol, but we added ALK vaccination, now you see that the tumor, they start 4 weeks, we treat with crizotinib, this is pre-treatment. We treat with crizotinib, tumors disappear. You see there are no longer tumors here. And still, after 16 weeks, the tumor, because of the vaccination, they are no longer back. So eventually, they come back later on. You see here from the curve. So at 16 weeks, there are still almost no tumor there, but eventually they start to come back after 20 weeks and follow up. But the difference between the crizotinib alone is pretty clear, and once more we could extend survival in this combination of treatment with ALK inhibitor and ALK vaccine. So we really thought that this is potentially a good way to combine a standard treatment now or a future standard treatment for ALK positive cancer, which is crizotinib inhibitor with an ALK vaccine. Next, we decided to try a different type of combination because very recently, last year, there was some evidence that some antibody that controlled the immune checkpoint could be effective against lung cancer. And what is the immune checkpoint? So now we are acting in this point of the immune reaction in which effector T cells, they recognize cancer cell, but the activity of these effector T cells can be changed or modulated by some receptor which are called CTLA-4 or PD-1. So if you give antibodies that block this receptor, which are inhibitory receptor, you can further boost the activity and the effector function of T cells. And so here is the cartoon in which you see that after T cell recognition, there are early inhibitory signal like CTLA-4 or late inhibitory signal, and you can really block this inhibitory signal on T cells by the use of specific CTLA-4 antibody or anti-PD-1 or PD-1 ligand antibody. So that was tested in these type of studies that I showed here, and they show that there is some efficacy, about 15–20% of response also in lung cancer. So our idea was why we do not combine ALK vaccine, ALK inhibitor, and this type of immunotherapy, immune checkpoint therapy, to see if we can further have more efficient cancer immune therapy. And that's what we did. First of all, we showed that ALK tumor, so this is an ALK tumor for our transgenic mice, compared to another model of EGFR mutated mice. EGFR is a typical mutation also in lung cancer, of KRAS mutated mice. Again, KRAS is a typical mutation in lung cancer. And we were comparing the expression of PD-1 ligand on the surface of this cancer cell, and we showed that ALK tumor, they express on average more than EGFR and KRAS tumors. So these were the basis to use anti-PD-1 ligand and PD-1 antibody to block this immune checkpoint. And the combination was by combining both an ALK inhibitor, in this case it's not crizotinib, it's Ty, but it is in the mouse system, it's an equivalent ALK inhibitor. We are doing the same experiment with crizotinib now, with this immune checkpoint antibodies in our protocol of vaccination. And these are preliminary data, but the response was positive. So at the beginning there were tumor. The tumor went down because of the ALK inhibitor treatment. So this is the decrease of size of tumor because of the ALK inhibitor here. And then we stopped giving the treatment with ALK inhibitor, and we were following up either vaccination alone, in this case, or vaccination plus immune checkpoint blockade. And you see that this combination is more efficient. Vaccination and immune checkpoint is more efficient in preventing the tumor relapse in the same site. Okay, the last part of the talk, the last, I would say, 5 to 7 minutes, will be on the resistant mechanism to ALK inhibitor in lung cancer. So there was almost immediate evidence by several papers that, as I anticipated before, the ALK inhibitor, specifically crizotinib, but that's true also for second generation inhibitor, that ALK inhibitor, they are very, very effective at the beginning for several months, but then eventually the tumor grows back because of several mechanisms or resistant to ALK inhibitor. And these resistance are clear, I would say, in at least 50, maybe 60% of the cases in which there is either an ALK amplification, so the fusion product, the translocation is amplified, so you require more inhibitor to treat, or single point mutation of ALK, I will be more specific later, or the bypass activation of other molecules, in particular other tyrosine kinase receptor that can bypass the shutoff of the ALK as a tyrosine kinase and compensate for its shutoff. So there is also, of course, a large number of tumors in which still the mechanism is unclear. So we wanted to focus because in the ALK amplification case, as well as in this bypass amplification, as I said before, in 100% of the case, the ALK expression is retained, so the vaccine is going to work, is predicted to work as well. But what about ALK mutation? Because ALK mutation, they really change, we can go here, they really change the structure of the ALK domain, so they can really change the immunological property of ALK as an antigen expressed by the cancer cell. So if now ALK is mutated, it can produce different peptide and be no longer recognized by a wild-type vaccine. And you see that the list of mutation is quite long. Many of these mutations were detected in lung cancer, but also some mutations in lymphoma and in inflammatory myofibroblastic tumor or neuroblastoma. And you see that there are several mutations that could potentially change the immunological property of ALK as an antigen in the tumor. So we decided to test few of them, mainly the most common one, which are the 1174, the 1196, and the 1156. These are the most commonly found mutations in resistant cases of lung carcinoma and other types of tumors. And you see, for example, the 11796 mutation affects an amino acid that is in the pocket that binds crizotinib. So when this amino acid is mutated, crizotinib is no longer able to bind efficiently the kinase domain of ALK, so it does not work anymore. So we decided to test this mutation by this assay. So we took a syngeneic bulk cell line, which I'll call S14, and in these cell lines, which are ALK negative, we express either an empty vector, which is here only with the GFP as a reporter to check the percentage of expressing cells. And then we transduce these cells either with the wild type ML4 ALK of the three most common mutations, the 1156, the 1174, and the 1196. So, and you see that the level of expression in this transduced cell line was pretty good and actually equivalent between the wild type and the mutated ones, and I would say very similar to the expression level in human cancer cell lines. So again, here is our referential expression for ML4 ALK in human cancer. So what is happening when you use this system? Then you take these cell lines and you can inject in the tail vein, and then these cell lines will home in the lung and will be grafted and generate several hundreds of tumors in the lung. So in tumors vaccinated with either control or ALK vaccine, if we were injecting cells without the expression of ALK, so these are ALK14 cells just transduced with the empty GFP positive vector, so there is no ALK in these cells, and either control mice or ALK vaccinated mice, you see there was no difference, of course, because the antigen is missing from the cells. So now there is no difference between control and vaccinated. But if we were expressing ALK in the grafted cells, now the difference is huge. You see that control cells, they still grow pretty strongly, about 200 tumors in 14 days. But in ALK vaccinated mice, now the number of tumors is minimal. There are actually very few and small tumors that you can barely detect by histology. So now tells that the ALK vaccine can recognize the ALK expressed by the cells and can block the graft and the growth of the tumors in the lung. And if we do the experiment with the three different mutants, as I said before, we got exactly similar results in which the ALK vaccine was able to block almost completely the growth of this mutant in the lung. And this is going to tell us that most likely this ALK vaccine is going to be active and very efficiently also against the mutated ALK tumor that developed after crizotinib treatment in human patients. So we think that, and I'm going to the final slides in which I summarize the points, that ALK elicited strong immune response also against lung cancer cells, reduced tumor growth, and then can be combined with ALK inhibitors, can be combined with immune checkpoint antibodies, and importantly is active against crizotinib-resistant mutants. And finally, this is the final slide in which we think that this could be potentially the future therapeutic strategy, in which we have any ALK-positive tumor. There will be a first phase of debulking of the tumor by chemotherapy or ALK inhibitor plus other type of inhibitor. And in the window of response, we can now treat the patient with an ALK vaccine, maybe with a combination of immunotherapy, check the efficiency of the response in terms of CD8, CD4, and eventually antibody response, even if these are dispensable, we showed before, and then eventually boost again the patient if the vaccination was not efficient, or follow up. And eventually, in case of relapse, there is still chances to treat a patient with second-line chemotherapy or different type of ALK inhibitor, and eventually do again the vaccination. So we envision that this could be a potential therapeutic strategy for ALK-positive tumor. So here are the acknowledgements. Most of the work was done both in my lab here in the Boston Children's Hospital at Harvard Medical School, and also some of the work, actually a good part of the work in the University of Torino in Italy, from which I come from. And of course, many of this data needed the help of clinicians at the Dana-Farber Cancer Institute of the Boston Children's Hospital as well. So now I think I thank you for listening, and I close my presentation here. I would be happy to answer some questions that if you want to pose me, and thank you very much. So maybe while I'm waiting for some questions, if there are, I go to the final slides, I guess, which is a reminder that if you want to obtain continuing education credits, you can go to this website that is listed here, and there will be specifically question and answer for this part of the talk to get this credits from this presentation.